My name is Ana Puhl and I am an Associate Director at Collaborations Pharmaceuticals, a company that specializes in developing treatments for rare and neglected diseases. Today, I will be talking about how we are developing treatments for rare diseases in a shoestring budget. And I'll be talking about the Batten disease CLN1 enzyme replacement therapy experience. This project started back on 2018, and so far we have raised 3.5 million in grants through the NIH. And this funding allows us to perform our preclinical studies, and right now we are moving to IND enabling toxicology studies, GMP manufacturing, and we are about to have our pre-IND meeting with the FDA. To move forward with this project, we will need to raise additional 6 million to progress to clinical trials. Only 700 out of 7,000 rare diseases have treatments, and this is due to lack of funding to translate to the clinic, and these therapies ended up in what is called the valley of death. Some big pharmas are increasingly working on rare diseases with larger populations because of the return of investments. Other companies focus on the latest therapies available, such as CRISPR and gene therapies. However, gene therapies are hard to develop for small companies, especially in this current economy. One factor that is important to highlight is that families make a difference in developing therapies for rare diseases, because the families, they fundraise, they start foundations, companies, they raise awareness, and they lobby politicians. And they contact the scientists and the community and help to make progresses on this field. There are many factors that are holding back the rare disease research. One of them is that some academic groups, they work in one disease, one protein, one target at a time. And some groups, they lack funding. They develop the therapies up to a point, and sometimes these therapies ended up being shelved and do not progress. Also, there is a need for an expertise to translate from preclinical work to the clinic and fill up the translational gap. One major important factor is the high cost associated with the developing of GMP manufacturing of proteins and gene therapy. Besides that, new modalities such as gene therapies, they can pose regulatory pathway challenges through the FDA. And all these factors, combined with the perceived lack of return of investments for rare diseases, can hamper the development, even when the priority review voucher is available and can be commercialized by about $100 million. There is an unmet need for rare disease treatments. The family foundations, they usually focus on patient care and early stages of drug discovery. Our company have taken on the challenge to develop an enzyme replacement therapy for CLN1. And why enzyme replacement therapy? Because enzyme replacement therapy has a proven record with the FDA. There are more than 10 enzyme replacement therapies approved for lysosomal storage disorders. Besides that, there are some advantages over gene therapy and the potential use for in utero treatment. Our company is taking a multi-pronged approach to developing treatments for rare diseases. And these approaches involve machine learning, where we can use machine learning to repurpose molecules for rare diseases. We also work on many diseases in parallel. Some of the steps can be scalable and can do in parallel, and also working on many diseases in parallel can help or increase the chance of finding molecules. We collaborate with experts in the field, and also when we need, we outsource to CDMOs, CROs, or other chemical synthesis when necessary. When we reach the point of having enough proof of concept, it's important to go to the FDA and get an early feedback, because that can save you some time and some money in the development. Besides that, our company leveraged the funding through the STTR and SBIRs through the NIH. As I mentioned, we use machine learning to discover new drugs for rare disease targets. And this approach involves curating the data, building machine learning models, generating predictions, and performing experiments. We have applied this approach to diseases such as sialidosis, chordoma, and more recently to CLN1. We work in many diseases in parallel, and this is important. Here I'm showing some of the diseases we've been working on, such as Batten, sialidosis, Pitt-Hopkins, and also some rare cancers, such as neuroblastoma and chordoma. But one factor when working with rare diseases that is important is to try to create or find a connection or a link with a bigger disease populations. In this case, the mutations on PPT1 protein that can cause the CLN1 disease, also that protein PPT1 has been shown to be overexpressed in cancer, and so that protein can be a target for cancers and also for aging. This enzyme replacement therapy project started because we knew that there was an enzyme replacement therapy that was developed at the university and was shelved due to lack of funding. So we wrote an SBIR and we collaborate with academic groups, with Dr. John Cooper and Dr. Mark Sands, and we perform our in vivo efficacy studies. Through those studies, we demonstrated that the intracerebral ventricular route was the best route to administer this therapy, and this treatment has statistically significant effects on gait, motor abnormalities, neuron loss, glial activation, storage material accumulation in CLN1 mice. This work was published, and also this data allow us to submit a phase 2 SBIR. Besides that, our work was also highlighted in a UK newspaper showing the gene-edited chip for CLN1 disease. Right now, we are executing our phase 2 SBIR grant. The goal of this grant is to manufacture this enzyme replacement therapy to perform IND toxicology studies and also to have our pre-IND meeting with the FDA. We interview and talk and got quotes with more than 10 CDMOs, and this was very important because the cost can vary widely. Like we got quotes from 2.5 million to 4 million, and we end up selecting STC Biologics for our cell line development, process analytical formulation, and manufacturing the protein that will be used in the IND toxicology studies. Our choice was based on a balance between price, expertise, and time. For our regulatory process, we end up selecting RTI International, and for the toxicology studies, Charles River. Right now, we're moving to the next step in our project, that is to have our pre-IND meeting with the FDA. This is a very important meeting where we'll be asking questions about our toxicology studies, if one species is enough, if our design covers the safety necessary to move and evaluate if the therapy is safe to move to clinical trials. Also, we'll be asking questions about our CMC process, our protein purification, and more factors involved in that development, and also some clinical endpoints, and if our clinical trial design is sufficient or is optimal. I have shown you how we were developing this therapy. This is our funding timeline. We raised more than 3.35 million in funding. The SBIR phase 1 in 2018 allowed us to perform our mice efficacy study. Later on, we got a diversity supplement that brought me on board to this project. We also participate in ICARP's business experience, and right now we are executing our phase 2 SBIR grant, where we're manufacturing the enzyme and we're start preparing our quality documents, preparing our pre-IND documents, regulatory, and also preparing for the GLP tox studies. The ICARP's experience was a 2-month intensive business bootcamp where we conducted 164 interviews with various stakeholders. We interviewed physicians, regulatory consultants, patients, CDMOs, and also we talked with families. And this was very important to give us insight was what is very important for the families, what they expect as a therapy. It helped us to identify some potential clinical sites, and also prepare us to move with our commercialization plan. And we were able to build a business model canvas for our project. As next steps, we will be executing this phase 2 SBIR grant. We will need to pursue additional funding through grants or VC funding. We will need about 6 million to make GMP protein, and also so 3 million for GMP manufacturing and 3 million for clinical trials. We need to focus on the clinical trials. We need to identify a PI for this project and the clinical sites. We will keep bringing additional consultants as needed, and we will continue to engage with CLN1 families and keeping them informed about our progress. One thing that's very important, we can scale our current business and repeat. We can develop treatments for other diseases in our pipeline using this approach. As a take-home message, here I show you how we develop a how we're developing a cost-effective therapy in a shoestring budget by leveraging the grant funding-based model for rare diseases. We hope that our approach inspires others to follow our footsteps and address the unmet need for more rare disease treatments, because we believe that no disease is too small. I would like to thank the amazing team of scientists that work with us in this project, and also our team at Collaborations Pharmaceuticals and the families and patient advocates. And a big thank you to NINDS for continuing supporting our research. Thank you.